Hello, good afternoon. My name is Ken Onel. I'm an associate professor of pediatrics at the University of Chicago. I'd like to thank you all for joining me today here in TV land, or computer land, I suppose. What I'm going to be talking to you about today is work that we've done in our lab looking at the role of genomics in trying to understand the etiology of cancer. As we go through the presentation, what I'd like to do is ask, as you have questions, you just feel free at any time to type them into the Q&A box, and at the end of the presentation, we'll go through them all together, and there you have it. So why don't we get started? So as I mentioned a few seconds ago, my laboratory's particular interest is in trying to understand how genetics contributes to cancer risk and response to therapy. As we all know, I'd like to, of course, mention I have no financial relationships to disclose, but with regard to our interest in cancer and the contribution of the genome to cancer risk, of course, we're not alone in that lofty goal. Our hope is to use the information that we learn to predict cancer susceptibility, to individualize cancer therapy, and to use genetic factors to identify new targets for novel rational clinical therapeutics. Now, as we all know, while that is, of course, the promise of genomic medicine and cancer, the reality hasn't lived up to that promise. And there are, of course, a number of reasons why. So here's an illustration of, of where we stand in terms of thinking about genetics in cancer. This is data from one of my absolute favorite studies performed by Doug Easton, Paul Farrow, and Bruce Ponder, looking at susceptibility to breast cancer in a very large group of British women, either with or without breast cancer. Ultimately, the study encompassed roughly 30,000 cases, women who developed breast cancer, and 30,000 controls, women who did not develop cancer. So on first blush, the study was a rip-roaring success. If you look at the p-values for SNPs that were identified as being associated with breast cancer etiology, I mean, we see numbers like 10 to the minus 76. In experimental science, of course, we should live so long as to see numbers like that. But when we look at the effect size, this victory is not so clear. So here, for example, we see effect sizes for the top SNPs, the most associated SNPs emerging from the study of 1.26, 1.11, 1.09, so on and so forth. And of course, what that means is that in the general population, if the risk of developing breast cancer is 1 in 100, should you be so unfortunate as to carry the SNP that's most associated with an increased risk for breast cancer, your risk goes from that 1 in 100 to a rip-roaring 1.26 in 100. So the question that has to be asked is, who cares? Why is it that the effect size of these SNPs is so small? So, people have tried a number of ways to sort of do better. One idea that people have had is, of course, to ask a bigger study. Another idea that people have had to sort of overcome the barriers to using genomic information clinically is to ask an even bigger study, but perform an even bigger study. But our idea is, rather than asking a bigger study, perhaps the right strategy is to ask a better study, a smarter study. The first step in performing a smarter study is, of course, to ask, why is it that the effect sizes that have emerged from these previous studies has been so small and so clinically of limited utility? While, of course, there are a lot of possible explanations for that, our task was to step back and see if we could come up with our own hypothesis as to why that was. And our reasoning for where we wound up is as follows. If you think of cancer as a complex disease, what that means, of course, is that it results from the actions and interactions of both genetic and environmental factors. If you think of these environmental factors, each of them, UV radiation, ionizing radiation, cigarette smoke, so on and so forth, elicits its own cellular response pathway, and each of those cellular response pathways has its own set of genetic determinants. And so we hypothesized that by not controlling for non-genetic noise, that is variation in etiologic exposures that are associated with cancer risk, all you're going to do is elicit genetic noise, essentially garbage in, garbage out. And the consequence of that, the inevitable consequence of that, is that a genetic signal would be attenuated. So our challenge was to come up with a method by which we could essentially control for that non-genetic heterogeneity, control for that non-genetic noise. And the strategy that we arrived at was to study second cancers, that is, therapy-induced cancers. The idea is that these therapy-induced cancers are a consequence of antecedent exposure to cytotoxic therapy. That exposure is the same for cases, that is, cancer patients who do develop these therapy-induced cancers, and controls, that is, cancer patients treated with the same agents who do not develop these therapy-induced malignancies. As a result, because the exposure is the same for the cases and control, non-genetic noise is essentially removed from the equation, and the only factors that are the major factors that distinguish cases from controls are their genetic differences. And so we hypothesized that by taking advantage of second cancers, we would facilitate our ability to detect genetic risk factors for cancer, because we would remove the confounding effect of variation in non-genetic factors. We thought that these second cancer genetic studies would provide a unique window into genetic susceptibilities for cancer. And because we're not looking at individuals or families that are genetically loaded, we thought that we might also learn something about gene by exposure interactions that are associated with risks for sporadic cancer. Now, the first study, our maiden foray, if you will, into the world of second malignancies, therapy-induced cancers, was a study we performed a couple of years ago looking at therapy-related AML, or t-AML. So therapy-related AML is a particularly devastating form of second cancers. Its survival is measured in months. Unfortunately, currently, it represents 10 to 20% of all cases of AML. It is cytogenetically characterized by two major subtypes. One that is associated with antecedent exposure to alkylating agents that comprises roughly 75% of all cases of therapy-related AML and is characterized cytogenetically by acquired abnormalities of chromosomes 5 and 7. It's also characterized clinically by a relatively long 3 to 10 year latency to the development of AML with an antecedent MDS phase. The other subtype of therapy-related AML is that is the subtype that's associated with antecedent exposure to topo II inhibitors. That constitutes roughly a quarter of all cases of therapy-related AML. It's associated with specific gene translocations, particularly involving MLL or AML1, and is also clinically associated with a very short latency. So one disease, two major subtypes. We performed a very small genome-wide association study, GWAS, looking at only 78 cases and 148 controls for therapy-related AML. Listed on this slide are our top 10 hits, top 10 independent hits. If you look at the rank, what you'll see is the numbers are not sequential, and that's because some of the top 10 associations were not independent. There was more than one SNP tagging a single locus. Okay. Now, as you might imagine, there's a reason, and we'll get to this, why 3 of those top 10 associations are highlighted, but we'll get to that in just a minute. So the University of Chicago set was our discovery set, so performed the GWAS in those 78 cases and 148 controls. We turned to our friends at Washington University in St. Louis for a replication set, which was comprised of 70 cases and 95 controls. the expectation or the hope when one performs these genome-wide association studies is that you would discover a set of associations and then replicate them in an independent second set of cases and controls. If you look at the University of Chicago set, what you see under the MAF columns are the minor allele frequencies for each of the 10 SNPs, first in controls and then in cases. And what you can see is that there's a very large difference that's reflected in a very small p-value. When, however, you look at the Washington University replication set, unfortunately, sadly for us, what you see is that while the control minor allele frequency is essentially the same as in the University of Chicago control set, the case minor allele frequency is very, very different and is in fact, in almost every case, very similar to the minor allele frequency that's seen in the controls. And so what that suggests is that for these top 10 associations, we were not able to replicate any of them. Naturally, we were upset and drank heavily for a while, but then of course we returned to the science. And when we actually looked at our University of Chicago discovery set of cases and the Washington University replication set of controls, you can see their clinical characteristics on this slide, but there was one thing that was particularly notable. If we look at the proportion of cases in those two sets that carried the chromosomal abnormalities of chromosomes 5 and 7, remember that those chromosomal abnormalities are associated with antecedent exposure to alkylating agents. What you can see is that they're vastly different. Now, if our original model is correct, that different exposures elicit different cellular response pathways with their own set of, each with its own set of genetic determinants, what this implies is that in the study, our discovery set and our replication set were in fact not comparable, but rather represented two different diseases, right, with different etiologies, because the cytogenetics were completely different in the two patient subsets. So what we did was we returned to our discovery and replication sets, and rather than treating all therapy-related AML cases as equivalent, what we did instead was we conditioned on the subset of cases for both the discovery and the replication set that had these acquired abnormalities of chromosomes 5 and 7. And here what you can see is that the results were very different. If we think back to the breast cancer genome-wide association study that we had discussed previously, it took 30,000 cases and 30,000 controls to come up with a handful of associations that were replicated. Now here, we took 10 associations, 10 SNPs, and attempted to replicate them in a second cohort. And if you look at the three SNPs that were boxed previously, it turns out that the boxes have moved, for which I apologize. But look at the three SNPs that are immediately below the boxes. And what you can see is that the case frequency, which had been very discordant in the overall data set, now by conditioning on patients that have the same somatic mutations, what you can see is that for SNP number 3, SNP number 5, I guess, and no, I'm sorry, for the RS1394384, I'll just read the numbers, for RS1381392, and for RS1199098, you can see that now, after controlling for differences in acquired cytogenetic abnormalities, suddenly for those three SNPs, the minor allele frequency in cases was essentially exactly the same in both the discovery set and the replication set. So our hit rate was 3 out of 10. Which is remarkable in a genome-wide association study. So we were very excited about that, and what that suggested to us was that, in fact, our hypothesis was likely to be true, that by conditioning on, in this case, surrogates of shared etiologic exposures, we would potentiate our ability to identify shared genetic susceptibilities. And in fact, that is the major lesson from this pilot GWAS that we had performed in therapy-related AML. By conditioning on exposures common to cases and controls, we can reduce the genetic noise due to exposure noise. We can enhance our power, paradoxically, even as the sample size is reduced. And what we can also do is use, in the case of therapy-related AML, shared driver mutations, such as acquired abnormalities of chromosomes 5 and 7, as surrogate markers for these shared etiologic exposures and therefore shared genetic susceptibilities. So after completing this genome-wide association study, we were encouraged to turn our hand to another second cancer and see if we could repeat our success or build upon our success. And in this case, what we did was we turned to second cancers in survivors of Hodgkin's lymphoma. So Hodgkin's lymphoma, as I'm sure most of you know, is a lymphoma. It occurs predominantly in the mediastinum, so the chest wall. The major method of treating patients with Hodgkin's lymphoma is radiation. And it turns out that the risk for second malignancies in survivors of Hodgkin's disease is associated with radiation exposure. And it occurs, the second cancers occur primarily in organs that are in the field of radiation. That would be, of course, the breast and thyroid, most commonly. It's important to note as well that the risk of these second malignancies is inversely associated with age of exposure to radiation. So patients with Hodgkin's lymphoma who are treated with radiation for their Hodgkin's lymphoma under the age of 20 are at a very high risk for the subsequent development of these second cancers, whereas patients who are treated with radiation for their Hodgkin's lymphoma over the age of 20 are at a much lower risk for the development of these second cancers. The study that we performed was undertaken in collaboration with a childhood cancer survivor study, the CCSS. This is a multi-institutional retrospective cohort study to investigate the long-term consequences of pediatric cancer and the effects of its treatment in survivors. The inclusion criteria for the CCSS is very simple. You have to have been diagnosed and treated for cancer between 1970 and 1986. You had to have been under the age of 21 at diagnosis, and you had to survive at least 5 years. To date, there are about 14,000 survivors who are still followed by the CCSS. This represents about 70% of the overall cohort, and underscoring the importance or the overrepresentation of second cancers in survivors of Hodgkin's lymphoma. It turns out that of the 802 second cancers that have been diagnosed in the CCS cohort to date, it turns out that 13% of Hodgkin's lymphoma survivors have developed these second cancers, whereas only under 5% of survivors of all other malignancies have developed these second cancers. So really, this is a radiation problem that is vastly overrepresented in survivors of Hodgkin's, and that was the rationale for our study. I also want to point out that second cancers in survivors of Hodgkin's lymphoma is not just an important or an interesting, to us at least, scientific problem, but it's one with an urgent clinical need as well, because it turns out that the second leading cause of death in survivors and patients with Hodgkin's disease is our second cancers, second only to relapse. So this is a big deal clinically as well as an important scientific question. So for our study, we were able to obtain germline DNA samples from almost 100 Hodgkin's survivors who did develop second cancers, and 82 Hodgkin's survivors who did not develop second cancers, but were all treated for their Hodgkin's lymphoma with radiation therapy. One thing I'd like to point out is that the latency for the development of the second cancers is quite long. It's 20 years. And so what we did was we required that all of our controls had survived at least 20 years plus one standard deviation, that is 5.5 years, in order to ensure that our controls were not future cases that had not yet declared themselves. Okay, so the idea was that by taking into account latency when choosing our controls, we would really minimize the likelihood that we would maximize the likelihood that we had a pure control data set. And this figure is a Manhattan plot. It shows the results of our genome-wide association study. For those of you who haven't worked with Manhattan plots, on the x-axis is plotted the position of each SNP that was genotyped in our study as a function of each chromosome. For this study, we did genotype almost 1 million SNPs throughout the genome. On the y-axis is plotted the negative log of the p-value of association. So the higher the dot is on the y-axis, the more significant is the association of that genotyped SNP and risk for second cancers following radiation therapy in Hodgkin's disease survivors. The red line that you can see at about 7 represents the threshold for genome-wide significance. So any SNP that surpasses that threshold is actually significant in our discovery set. And what you can see is that there are 3 SNPs that surpass that threshold, 2 on chromosome 6 and another one on chromosome 18. Now, as with the therapy-related AML GWAS, the bar in these genome-wide association studies is not just to discover, but to replicate. And so what we did was we put together not one, unfortunately, again, there's a formatting issue with this slide, for which I apologize. We put together 2 replication sets, one keeping in mind that risk for these second cancers is much higher in individuals who are radiated under the age of 20. And the other so that the first control replication set was individuals who are treated for Hodgkin's with radiation under the age of 20. And the second is comprised of individuals who were treated with radiation for Hodgkin's lymphoma over the age of 20. And then what we did was we tested the association of each of those top 3 SNPs, the ones surpassing the threshold for genome-wide significance, separately in each of these 2 replication sets. And the results are shown here. This is a representation of what are called forest plots. The red and blue lines represent the 2 chromosome 6 SNPs. The green line represents the chromosome 18 SNP. And what you can see in the upper right-hand corner is the RS number of each of the 3 SNPs themselves. Okay. If we look first at the bottom of the slide, what you can see is that so the lines themselves, the thick part, the square box represents the actual odds ratio of association. And then the 2 lines spreading out from either side represent the 95% confidence interval. If you look at the x-axis, the odds ratio is plotted, and any association that touches 1, an odds ratio of 1 is by definition not significant. So if you look at the replication cohort of individuals who are irradiated over the age of 20, the bottom box, forest plot, you see that none of the 3 SNPs was significant. On the other hand, if you look at the replication set for individuals who are irradiated for their Hodgkin's lymphoma under the age of 20, what you can see is that the 2 SNPs that are on chromosome 6 are both significant. So what was really cool was that we had discovered and replicated 2 associations, both on chromosome 6, that were associated with a predisposition to second cancers following Hodgkin's lymphoma. As we'll sort of underscore in the next slide, if you look at the odds ratio in these replication sets, the numbers aren't 1.09, 1.11, 1.26, but are actually much greater, 2.52, 1.69. So as you would expect, in these small sets, the odds ratios have to be very great for them to be detected and detected significantly, but in fact we were able to achieve that. What was really cool is that recalling that risk for second cancers is greatest in those irradiated under the age of 20. Our genetic results reflect that epidemiologic observation. We have an association that was discovered and replicated in individuals who were treated under the age of 20, but nothing in individuals who were treated over the age of 20. So that was a really nice sort of genetic echoing of the epidemiologic data. If we look in the combined discovery and replication set here on this in this graph is shown the odds ratios for each of the two SNPs on chromosome 6, and you can see 3.21 and 2.43, which are of course astronomical effect sizes to emerge from a genome-wide association study. So the next question, of course, is what are these SNPs and where are they? So here is a blow-up of the region of the two SNPs on chromosome 6 that shows both directly genotyped in the orange and imputed in the black SNP, in the black SNPs in the region of chromosome 6 Q21 that's tagged by the two SNPs that emerged from our genome-wide association study. Again, if you look at the dotted line, that's the threshold for genome-wide significance. What you can see is that the two SNPs that were directly genotyped, the RS494 SNP and the RS104 SNPs, are the two that had the highest degree of association with second cancers following Hodgkin's lymphoma. And what you can also see is that the two SNPs are themselves very close to each other. It turns out that the two SNPs are roughly 4 kilobases apart from each other. They're in moderate LD with an R-squared of about 0.4. They live in a region intergenic between two genes, PRDM1, which is also known as BLIMP1, and ATG5, which is an autophagy gene. That intergenic region is itself roughly 70 kb. And if you sort of look at the peaks, what you can see is that the two SNPs and the large number of other associated genotype and imputed SNPs are also both within a single recombination block, which defines groups of SNPs that travel together. Okay? Now, when we conditioned on one SNP, we found that there was a residual association remaining for the other SNP. And that's important because what that indicates is that the two SNPs together tag the true functional variant better than either SNP does alone. And so what we did was we decided to perform functional studies to see if we could get at what the mechanism by which these SNPs were associated with radiation carcinogenesis. And we did so by creating a haplotype of the two SNPs, since they together provided more information than either alone. What we did was we defined the risk haplotype as the haplotype that has both risk SNPs, okay? And we defined the protective haplotype as having both protective alleles, okay? So risk haplotype, both risk alleles, the protective haplotype, both protective alleles, okay? So then the first thing that we did was we asked whether these SNPs were associated with gene expression for any gene that was within 10 megabases of the two SNPs. And the rationale is that when genome-wide association studies were initially done, the hypothesis was that the SNPs that would emerge would be SNPs that changed amino acids. It turns out that that's not the case. It turns out that most of the SNPs that are emerging from GWAS studies function by altering transcription. So they either change levels of a message or they somehow affect enhancers or some other function like that, like enhancer repressor functions or alternative splicing forms of genes, of transcripts. And so to test whether or not our SNPs were likely to be an enhancer for PRDM1, ATG5, or any other gene within 10 megabases in either direction of the two SNPs, we took a data set of expression for which we also had full genotype information, and we asked whether the haplotypes, risk, protective, or intermediate, homozygous risk, homozygous protective, or intermediate, were associated with levels of mRNA for any of the genes within that, as it turned out, 20 megabase window. And it turns out that the haplotype was associated with expression of only one gene, and that is PRDM1. And what you can see here is that in individuals who had increasing dosage of the risk haplotype, they had lower, significantly lower expression levels of PRDM1 mRNA. Okay, so what that means then is that the haplotype is an EQTL, an expression quantitative trait locus for PRDM1, okay, with more risk equals lower transcript, okay? So that's great for message, but the question is, does it have any meaning for the protein? And we'll get to that in a second, but before doing that, if in fact our haplotype represents an enhancer, one would expect then that we could see that by looking at levels of unprocessed transcript in the context of the protective haplotype. And so that's what we did. What we did was in the red, you see steady state mRNA levels for PRDM1 mRNA at variable times following irradiation. In the blue, you see levels of the primary transcript, so the unprocessed transcript, following radiation. And what you can see is that in the context of the protective cells homozygous for the protective haplotype, there is a significant increase in primary transcript following radiation, that echoes and precedes levels of the steady state transcript, arguing that the region tagged by our protective haplotype tags an enhancer for PRDM1. Although it's not shown in this slide, when we do the same thing in cells homozygous for the risk haplotype, there's no change in PRDM1 message levels, either primary transcript, unprocessed transcript, or steady state mRNA. And so taken together, what those observations indicate is that the protective haplotype is indeed an IR-inducible, tags an IR-inducible enhancer region that regulates PRDM1 transcription, which is really exciting and interesting, okay? So now let's ask the question, what happens to the protein, right? That's message, is that echoed in protein? And so to test this, what we did was we took 3 cell lines that are homozygous for the protective haplotype and 3 cell lines that are homozygous for the risk haplotype. We either left them alone, growing exponentially, or we treated them with radiation and then looked at levels of PRDM1, ATG5, remember that was the gene on the other side of the haplotype, and just as a loading control ran. And what you can see is a couple of things that are incredibly interesting, right? First, what you can see is that baseline levels of PRDM1 is much higher in the context of the cells homozygous for the protective haplotype than in cells homozygous for the risk haplotype. Second, you can see that following radiation, although this gel is a little bit blown out, you can see that there is a significant induction of PRDM1 following radiation in cells homozygous for the protective haplotype, whereas there is no induction of PRDM1 in cells homozygous for the risk haplotype. If we look at ATG5, we see nothing happens, and of course, if we look at our loading control, we see that there's roughly equivalent loading in this gel throughout the experiment. Okay, so if we then move to the next slide and expand that experiment to not 3 cell lines, but rather to 4 cell lines, and then plot out the data, we see quantitatively the same thing. So first, if we look at cells, the pink box, that represents the 4 cell lines homozygous for the protective haplotype. The blue boxes are cells that are homozygous for the risk haplotype. And what you can see is that in cells homozygous for the protective haplotype, first basal levels of PRDM1 are significantly greater than in cells homozygous for the risk haplotype. And then you can see that following radiation, whereas PRDM1 levels rapidly increase and remain significantly increased through at least 4 hours following irradiation in cells that are homozygous for the protective haplotype. There is no induction of PRDM1 in cells that are homozygous for the risk haplotype. So what we see is that following radiation, PRDM1 in the context of the protective haplotype is radiation inducible, but it's not so in the context of the risk haplotype. Okay, so, and this is important and interesting. It echoes the EQTL data because remember, the risk haplotype was associated with lower levels of PRDM1 expression, and we see that that's also the case in the level of protein. So that's great, but what does that mean in terms of radiation breast carcinogenesis? Well, it turns out that a gene that is commonly altered in radiation breast carcinogenesis. Remember, breast is one of the most common types of second cancers in survivors of Hodgkin's lymphoma. In these patients and in cell lines that are designed to reflect the radiation carcinogenesis paradigm, what we know is that MYC is often significantly altered. We also know that the major function of PRDM1 in the cell is to act as a transcriptional repressor. And in fact, one of the targets of PRDM1 is MYC. So we hypothesized is that MYC would be differentially regulated by PRDM1 in the following radiation in the context of cells that are either homozygous for the risk or homozygous for the protective haplotype. And to test this, we again took 4 cell lines, homozygous for the risk, 4 cell lines homozygous for the protective haplotypes, treated them with radiation, and in this case, we blotted for MYC. And what you can see, once again, pink representing the protective cells homozygous for the protective haplotype, and blue representing cells homozygous for the risk haplotype, is that following radiation, there is a rapid and very significant repression of of MYC in cells that are homozygous for the protective PRDM1 haplotype. In contrast, the repression of MYC is slower and less significant in cells that are homozygous for the risk haplotype. So what we've been able to do is mechanistically go from a genome-wide association study in which we identified a haplotype to the function of that haplotype in differentially regulating PRDM1 induction following radiation to actually showing that that matters for a gene that's involved in radiation carcinogenesis, that is MYC. So that's really cool and really sort of takes the study from beginning through the mechanism. So having done that, our next question was to ask, what is the signal transduction pathway from radiation to PRDM1? Now, we know that in other contexts, PRDM1 is induced by NF-kappa B in the context of other forms of cellular stress, such as ER stress. So we hypothesized that perhaps NF-kappa B was also a critical inducer of PRDM1 following radiation. And to test this, we again took cells that were either homozygous for the protective haplotype, here in blue, or homozygous for the risk haplotype, here in red. We left them either untreated, which is the first 2 columns, and we see the expected differences in PRDM1 baseline levels. We treated them with radiation. We see the induction of PRDM1 in cells homozygous for the protective haplotype, but not in cells homozygous for the risk haplotype. And then in the final pair of bars, we pre-treated cells with a chemical, very specific NF-kappa B inhibitor and then treated the cells with radiation. And what we observed was that NF-kappa B blockage completely abrogated and prevented PRDM1 induction by radiation, suggesting that the signal transduction pathway from radiation to PRDM1 involved NF-kappa B signaling. So we then turned our attention to trying to identify the actual SNP that was tagged by our haplotype. This figure over here is a blow-up of the region that is tagged by the haplotype. The green box shows you the PRDM1 gene region itself. The region that is downstream of that is the entire region that's tagged by the haplotype. And to identify, we know that there's an enhancer that's tagged by the haplotype. Our first hypothesis was that it would be something that altered an NF-kappa B binding site. And while, in fact, there are several NF-kappa B enhancer regions that are enhancer sites that are in the region that are tagged by the haplotype, there's no genetic variation that exists in any of them. So what that means is that it's highly unlikely that our haplotype tags a SNP that's in an NF-kappa B enhancer region. So what could it actually be? Well, we used a variety of bioinformatics tools to try to winnow down our choices in that relatively large 40 kb real estate region. We looked at marks of DNA's hypersensitivity, marks of open chromatin, RNA Pol II binding, evolutionary conservation, and we sort of summed that information together over the region, and that led us to identify four candidate enhancer regions that might be tagged by our haplotype that we refer to as E1 through E4. Sort of in conjunction with that analysis, we also performed an integrative bioinformatics analysis that we hoped would lead us to the actual functional SNP. So here, what I plotted for you using the 1000 genome database is all of the genetic variation that is tagged by our haplotype. What we have plotted here to orient you is the negative log p-value of the association between all of those SNPs and risk for second cancers following Hodgkin's disease. The two SNPs that are marked in the blue are the two SNPs that surpass the threshold for genome-wide significance in our GWAS. It turns out that this very same region has been identified as being associated with both rheumatoid arthritis and lupus, both of which, of course, are B lymphocyte disorders as well. So invoking the concept of pleiotropy, that is, things converge on the same pathway or the same SNPs, we hypothesized that perhaps the same SNP was associated with both RA and lupus and with a predisposition to second cancers following Hodgkin's disease. And so here are the SNPs in that region and their LD, their linkage to equilibrium, with the most associated SNPs from either the lupus GWASs or the rheumatoid arthritis GWAS. Now, in this, and we've also shown here the regions of open chromatin in this small genomic region. Now, the reason that we did this was because we expected, remember, tag SNPs only tag a region. A functional SNP, if we're right, should be associated with all of the different GWAS studies and should also exist in a region of open chromatin. We were able to take advantage of the fact that one of the genome-wide association studies, one in lupus, had been performed in a Chinese population, and Chinese and Caucasian individuals have different LD structures, and so that we hypothesized would help us winnow down the possible candidate SNPs that might actually be tagged by our locus. And so the first thing that we did was we observed that the two candidate enhancer regions E1 and E2 sort of existed in between regions of genetic variation. So there were no SNPs that are in either E1 or E2, so those are gone. If we look at E4, while we see that the association between while we see that E4 exists in a region of open chromatin, we see that there are no SNPs that are associated with lupus in the Chinese population. And what that suggests is that, in fact, enhancer 4 doesn't contain the functional SNP because, again, functional SNP should cross ethnicities. If we look, however, at E3, what we see is that there's one SNP that is highly associated with all the different diseases and all the different ethnicities and lies in a region of open chromatin, and that's marked by this arrow. If we ask what that SNP actually does, we see that it lies, the RS115 SNP, we see that it lies in an sp3 binding site. sp3 is a well-characterized transcriptional repressor, and if we look at open chromatin, what we see, and again, this is shifted somewhat from its original version, what we see is that the SNP itself lies in region of open chromatin and at a site for RNA Pol II binding. So taking it all together, we hypothesized that enhancer 3 was likely to contain the true functional SNP tagged by our haplotype, and that SNP itself was very likely to be the 115 SNP. To test this hypothesis, we first performed a luciferase reporter experiment to look individually at the 4 different candidate enhancer regions. You see here the plasmid vector that we used for these studies. So there's an SV40 promoter driving luciferase expression, which you can just measure experimentally. What we did was we cloned into that BAMH1 site that you can see each of the 4 different candidate enhancer regions. The hypothesis is that a functional enhancer is going to alter luciferase expression. And what we saw was that only enhancer 3, the one that we had predicted informatically was functional, did in fact alter luciferase expression, suggesting that the functional SNP tagged by our haplotype in fact does lie in the enhancer 3 region. Suggesting that in fact the SNP itself that we identified in the sp3 binding site was functional. We looked at RNA Pol II binding across the region of the SNP, and what you can see is that the TT represents cells that are homozygous for the protective version of that SNP. The CC represents cells that are homozygous for the risk version of that SNP. RNA Pol II binds to regions of active transcription, so enhancer and promoter regions. And what you can see is that exactly what you would predict, that there's increased RNA Pol II binding in cells homozygous for the protective version of the SNP, and there is decreased RNA Pol II binding in cells homozygous for the risk version of the SNP. And this leads us to 2 conclusions. First, it leads us to conclude that in fact this SNP is truly a functional SNP, okay, so it actually matters. And the second thing is that the RNA Pol II binding echoes what we would have predicted for a SNP that sits in a repressor region, okay, in that there is less expression emerging in cells homozygous for that sp3 site than there is in cells that have destroyed the sp3 site, okay? Then the final experiment that we did was, again, looking at function. We performed an electromobility shift assay in which we took an oligonucleotide that contained either the risk allele or the protective allele. We washed it with protein extract, and then we asked about the binding of that protein extract to to the labeled version of our oligonucleotide probe. And what you can see is, first, if you look at the bar graph on the right-hand side, the protein's nuclear extract bound more avidly to probes that contained the risk allele than to probes that contain the protective allele, suggesting that in fact the nuclear extracts contain some sort of transcriptional repressor, right? And then what we can see is that when we washed off the binding by competing it off with unlabeled probes to sort of draw off the nuclear extract, that there was it was more difficult to draw off the nuclear protein from cells that from probes that had the risk allele than that it was more specificity when we used probes, unlabeled probes that had the risk allele than unlabeled probes that had the protective allele, suggesting that not only was the binding of this nuclear protein to the risk oligonucleotide, not only was it more avid, but it was also more specific. Okay. And so again, that suggests that this is truly a functional SNP. So now, taking all of these data together, what's our model? Our model for how our SNP actually, or how our haplotype actually is involved in response to radiation, is that in the context of the protective allele of the RS111 SNP, the sp3 binding site, sp3 cannot bind to that enhancer site. Rather, RNA Pol II and NF-kappa B can bind, and they elicit transcription of PRDM1. In the context of the risk allele, remember, we generate an NF-kappa B, an sp3 binding site. sp3 binds to that enhancer site, preventing Pol II and NF-kappa B from binding to the enhancer and the promoter, and thereby abrogating transcription of PRDM1 in response to both baseline or limiting in response to radiation, and also both at baseline as well. Okay, so that's how we're thinking that our SNP actually, or our haplotype actually, is associated with radiation carcinogenesis. So the question then becomes, this is great in second cancers where there's this very large and very focused exposure to radiation, but does it have any relevance for sporadic disease? So this is actually difficult to test and clinically very important, since, as we all know, radiation exposure is ubiquitous. So what we did to test this was we turned to a collaborator, Yuri Nikiforov, who has been studying for a number of years survivors of Chernobyl. And what we did was we asked whether one of the SNPs that was associated with radiation-induced second cancers was also associated with risk for radiation-induced thyroid cancer in survivors and children who survived Chernobyl. And the point that I want to make is that while this sample set was extremely small, what we observed was an effect size that was in the right direction and almost achieved statistical significance, suggesting that the region that we identified may be involved not just with risk for radiation-induced second cancers, but with risk for radiation-induced primary cancers as well. So what is the meaning of all of this? Why is it that we spent all of this time doing these sorts of studies? Well, the answer has to do, of course, with cancer susceptibility. Right now, so as well as being a researcher, I'm a clinician, and my clinic is dedicated to familial cancer, trying to help families that may have an increased risk of cancer understand why that is, and to help them try to identify perhaps strategies by which they can minimize their increased risk of cancer. The problem is that right now we have very few tools that we can use to help us in this very important task. We have only family history of cancer and personal history of cancer. And because families are small and because people don't necessarily know their family history, and because it's there are lots of reasons that people die other than cancer, we are very limited in our ability to actually utilize these clues to perform clinical risk assessments for families and for cancer risk. And we can only identify very highly penetrant cancer risk susceptibility genes such as BRCA1 or BRCA2, of course, associated with predisposition towards hereditary breast and hereditary ovarian cancer. So these are blunt objects, very, very crude measures of assessing cancer risk. If we think about the results of our GWAS, I want to sort of turn it around from presenting the data as odds ratios to presenting the data as something that I think is actually much more clinically relevant, and that is genotype-specific penetrance. And what I mean by that will become clear in just a second. If we look at the first box, the orange box, what we see is that overall, by 30 years following radiation, roughly 18.5% of survivors of Hodgkin lymphoma will develop a second malignancy. If we look at individuals who are homozygous for the protective haplotype, it turns out that by 30 years following exposure to radiation, only about 3% of individuals have developed a second malignancy. In contrast, if we look at individuals who are homozygous for the Risk Haplotype, what we see is 30% of them have developed a second cancer. So 3% versus 30%, that is enormous. And that, to me, is a very important clinical clue. I mean, the penetrance of our haplotype is almost BRCA2 level penetrance. The important point, two important points are: one, it's only in the context of radiation. So if you look at the results of a GWAS of just breast cancer, you'll never see our SNPs emerging, not even at a nominal p-value threshold of 0.05. And second, look how common these SNPs are. It turns out that 50% of Europeans are Caucasians are homozygous for the Risk haplotype. And so what that means is that they're very, very common, very, very penetrant, but only in the context of a specific exposure. So context matters, right? So I think this has a lot of potential implications for the way that we think about cancer risk. And I think that one of the important implications is that it may suggest that we may be able one day to go from thinking of cancer risk as we do right now, basically one size fits all. We have these very few, very blunt objects that we can use to think about cancer risk, to hopefully in the near future be able to do something that's a little bit more precise. But what we can do is think about genetic susceptibilities in an exposure-driven way. Oh, you are highly predisposed to develop cancers following radiation exposure, and so your risk prevention strategy should be avoid radiation. And we can do this sort of analysis or use this sort of strategy for a variety of different exposures, and I hope that we'll be able to then achieve a more subtle method by which we can assess an individual's risk for cancer. So with that, as our time is drawing to a close, I think I too will draw to a close. Our conclusions from the study are: first, a hypothesis-driven GWAS can be successfully performed, and I think that's an important point because one of the big criticisms of these genome-wide association studies is that they're hypothesis-free and at the same time very expensive. And so because they've had such limited the results have had such limited clinical utility, the question is, why should we bother doing that? So in fact, I would argue that we can do hypothesis-driven GWAS, and the results can be quite powerful. By taking into account exposures, we can see that these exposures are important drivers of genetic associations that are associated with these specific malignancies. We've seen that common variants can have very large effects, but only in the right context. And conversely, we've touched on the fact that variants, even with very large effects in the right context, can appear neutral when that context is not taken into account. Based upon the data that we've discussed, I would argue that we can conclude that PRDM1 is a radiation-responsive tumor suppressor. PRDM1 is associated with radiation-induced cancers following radiation-induced carcinogenesis. It's induced in individuals that have the protective haplotype, but not in individuals that have the risk haplotype. And that haplotype differentially regulates, a key mediator of radiation carcinogenesis, that is MYC. One other take-home point that I hope has come through is that for studies such as these, because they're predicated upon a shared exposure, the sorts of follow-up experiments that we've discussed today readily present themselves. We know that this is a radiation phenotype, so take cells of opposing genotype, irradiate them, see what happens. And then the final point that I want to make is that I hope that ultimately integrating these sorts of G/E interactions into genetic studies may lead us to a time when we can use this information to contribute to clinical decision-making. As an example, should an adolescent woman with Hodgkin lymphoma be treated with radiation therapy? I think is an important question that we may be able to address in the near future. So I'll stop there. I want to thank the people in the lab and our collaborators who actually did the work. The work that I described, gene therapy-related AML, was performed by Jeff Knight. Sapna Vora, who's a current grad student in the lab, has been following up on his studies. The second cancer after Hodgkin lymphoma studies were performed by Timothy Best, a superb graduate student who recently finished his time in the lab. The CCSS were wonderful collaborators for this, and so I thank Les Robeson, Smita Bhatia, Louise Strong, and Stella Davies, who are the leaders of the CCSS. And we had a variety of collaborators, including Wendy Kozin from USC and Andrew Skoll, who made all of this possible. We couldn't have done it without their help, and also without the help of my close friend from Newcastle, James Allen. So I'd stop there. I thought that you guys should actually see the lab. This is us. This is from our brief foray into musical theater. As you will be relieved to know, we were forced out of musical theater and sadly have returned to science. So thank you for your attention. If you are interested in obtaining CME credits for this talk, the website is here on the screen. And at this point, I'll be happy to take any questions. So I see that the Q&A box is blinking. Let me open it up. I just see Dr. Onel. Thank you for your presentation. This is from Darlene. I believe I don't see a question that's attached to that. So if there are any questions, this would be a good time to enter them. Thank you, Darlene. I am glad that you enjoyed this presentation. If there are any other questions, I'd be happy to take them now. Well, it appears that there are no further questions. So I guess with that, once again, I thank you for your attention. And I will, I guess that's it then. Thank you. I guess we can log off now.